Good morning, everyone. Good morning. Good to see you. Good to see you on this very gloomy morning. Yes, please go ahead and sign into chat for me. Apparently, we're due for a lot of rain again today, at least this morning. I hope everybody's staying dry. Good to see you. I've missed you. I haven't had a chance to see your smiling faces. Very good. So today we're going to be talking about microbial metabolism. And specifically, we're going to be talking about carbohydrate metabolism. I wanted to say just a couple of things about your lab practical exam. Um, let's start there. If you haven't already, take a look at your lab practical 2 grade when you have a chance. Open up the assignment, click on the link, and take a look at the comments that I may have made on your exam. Overall, the grades were good. I was very pleased. But you may have noticed that your grade dropped a little bit compared to the first practical. Again, I don't think anybody's grade dropped a whole lot, but pay attention to that. Pay attention to the fact that that lab practical was more difficult than the first lab practical. And you have another lab practical. You have the third one to do as well. That one's going to be the hardest. That one's going to be the hardest one. So if you had some issues on this lab practical, if you felt as though your notes were not complete enough, be sure to address that now. Be sure to go back through and make sure that your lab notes are as thorough as you can make them, so that they will be an excellent resource for you while you're taking that third lab practical in a couple of weeks. There was no one particular question that students had trouble with, so I won't talk about any one particular question, but I will mention one sort of a strategy that it's worth reviewing together as a class. Um, Compared to the lecture exams, of course, the lab practical exams are questions where you have to provide a short answer or a short essay. And sometimes it's very easy. Sometimes I literally ask you yes or no, top or bottom, left or right. But other times I ask you to explain something, or I'll ask you the question, how does something happen? Or why does something happen? It's a really good strategy for any examination when you have to answer a question in a short answer or an essay. When you see those terms, when you see the term explain, or how does this happen, or why does this happen, that's a signal to you as a student that your instructor wants you to show them what you know. So, for example, there was a question on the practical about ultraviolet radiation and how it damages microbial cells. That was an opportunity for you to explain in detail, in specific detail, what you know about ultraviolet's impact on the DNA molecule. And many of you did a great job, a great job. You told me everything you knew about ultraviolet and DNA. You even went on to tell me that Well, you told me that thymine dimers get created, and you explained to me that those break the double helix. And many of you went on to explain that that can even cause more problems when the time comes for DNA replication to occur. That was excellent. Some of you wrote really nice, detailed, specific answers for that question. So watch for those words. Watch for those words, explain, or tell me how, or tell me why. Those are open-ended terms. And when an instructor uses a question like that or a term like that on an exam, it's your invitation to be specific and complete, to tell them what you know in detail. Okay, just a little strategy there. Um, Everybody can use good strategies when they're facing exams. All right, so lab practical 3 will be your last lab practical. That's coming in a couple of weeks. And that one's going to be tough because that one will cover the last set of labs, and they are the toughest labs because they are packed with information. And they're also packed with little experiments and results and how to interpret those results. So it's really important that you are on the top of your game taking notes during lab periods. Okay? And if you have to miss a lab or if you have to leave lab early, you've got to go back and watch the video. I promise you, if you don't, you're not going to do well on the lab practical. And I would hate to see your grade come down after you've worked so hard. I would hate that for you. So if you need to do it, go back and pull up some of the videos and fatten up your notes. Fatten up your lab notes. Go through and listen again to things that maybe didn't make sense to you the first time, or things that you didn't quite get enough notes for. It's worth your effort. It's worth your effort. It will pay off. The other thing I'll mention is this. You have your next lecture exam next week, and that lecture exam is a tough one. That lecture exam covers 4 individual topics, even though it says 2. It says microbial metabolism and microbial genetics. But it's really 4 topics, right? Because it's enzymes and energy, it's cellular respiration, oxidative phosphorylation, and fermentation. It's DNA replication and mutations, which you'll be learning about this week. And the last one, which is gene expression, transcription, and translation. 4 very difficult topics. So again, same thing with your lecture notes. Give those lecture videos the time that they need. Make sure that you put aside even extra time this week to get your lecture notes up to snuff. The exam that you're having next week is much more difficult than the exam you had, 5 or 6 weeks ago. So don't fall into the trap that some students fall into of thinking, well, I did really well on exam 1, so I don't need to put too much work in this week because I did really well on exam 1 and I did well on exam 2. Well, this exam is a whole different animal because you know a lot now about microbiology. You've got a lot of information now in your arsenal. And again, these topics that you're learning last week and this week are the hardest things, as far as I'm concerned, the hardest things we cover: microbial metabolism and microbial genetics. And again, some of you have a very strong background and others of you don't. And if you know you don't have a strong background, in how cells use DNA in the processes of transcription and translation. If you know that you never really understood that very well, or you're really not comfortable with the terminology and the language, that's your signal to put the extra time in this week. I don't want to see all your hard work and your excellent grade go down now, because we're at the most difficult part of the course. All right, fight for it. Fight for your good grade. All right. So I think that's all I wanted to mention. Feel free to type questions into chat if you have any. But as I said, do take the time to pull up your lab practical and read any comments I might have made for you, because sometimes if you get a question wrong, you may have had something in your notes that you need to adjust a little bit. You need to add to it or take away from something. And sometimes my comments are geared to that. You may have had a little misunderstanding, or maybe there was a detail that you didn't have in your notes that you should have in your notes. Okay. All right. All right, very good. So I'm just going to quickly pull up our canvas for this week. Give me just a sec. Let me try this again. So you should see on your screen that I'm in our course modules. Here's the week ahead. Your to-do list, microbial genetics, DNA replication and mutations, microbial genetics, transcription and translation. Those are the two processes that make up what we call gene expression. This is how genes get expressed, through transcription and translation and the production of protein. So today we're going to talk about carbohydrate catabolism in lab, and on Wednesday we'll talk about fermentation. Now you can see that there's a little supplement document here called the catabolism of glucose. I'll explain that one a little bit more during lab today. And here are your lab questions. Okay. All right. So another busy week. Yeah, I hope everybody is staying dry. We've had an awful lot of rain, haven't we? It's good. It's a good thing. We need it, but my goodness, it's been raining. All right, I'm going to pull up our lab slides. And that's what you should be seeing on your screen now. The title slide for carbohydrate catabolism. So here's our objectives for today. First, we're just going to review some of the terminology around carbohydrates. Then we're going to talk about the differences between oxidative catabolism and fermentative catabolism. Now I'm saying those words slowly because this is where the rubber meets the road in terms of difficult language. It's critical that you put together a vocabulary list for yourself of any terminology that you are not really comfortable with, so that when you see terms like oxidative and terms like catabolism, you feel very comfortable with them. For our exercise today, we're going to perform and interpret two different tests. One is called the Starch Hydrolysis test, and one is called the oxidative fermentative test, or what's most commonly called the OF test. So let's review some terminology around carbohydrates, because this can become very confusing. Remember, in science, the term sugar refers to a carbohydrate. We use those two terms interchangeably. Now, this can be problematic when you're talking to people who are not students of science, because when you say the word sugar to a layman, they think about sucrose. They think about table sugar, white sugar. All right. There are many, many, many carbohydrates, not just table sugar. Okay. So yes, scientists will use the word sugar interchangeably with the word carbohydrate. But we have to remember that most people, when they hear that word, they think of the white crystallized sugar that they put in their coffee each morning. So within the carbohydrates, within this class of macromolecule, there are different types. There are monosaccharides, there are disaccharides, there are oligosaccharides, and there are polysaccharides. That term saccharide translates as sugar. So when you see the word monosaccharide, that translates to one sugar. And the way you should interpret that is, that means that that's a carbohydrate that has one subunit in it, one sugar. Now, the other term that you can use interchangeably with the term monosaccharide is simple sugar. And you'll hear that word a lot in common language. You'll hear that phrase a lot. People always talk about simple sugars in the diet. Now, what are some examples of monosaccharides? The big one for us in microbiology, and really in all of metabolism, is glucose. All right, glucose is the central monosaccharide that living cells use to produce energy, to produce that molecule we've talked about called ATP. Now, you should also know, since you're all interested in healthcare, that the word dextrose is often used as a replacement for the word glucose. So those two words are interchangeable. Another simple sugar is the one called fructose, and that's the fruit sugar that we're all familiar with. Now, if mono means one, the term di means two. So a disaccharide is a molecule that's made out of two sugars or two subunits. And one important thing to note is that in nature, in the disaccharides that living cells would make use of, one of the subunits is usually glucose. So, for example, we talk sometimes about lactose in here. I mentioned sucrose a minute ago. There's one called maltose. Those are disaccharides. And for each one of those, one of the subunits is glucose. So lactose, which is often called milk sugar, that's composed of one molecule of glucose bound to one molecule of galactose. And those two simple sugars together, or those two single sugars together, make up this disaccharide. Sucrose, table sugar, is a combination of glucose and fructose. And maltose is a combination of two glucose molecules. All right, disaccharide, two sugar units bound together. Now, this is where it gets a little bit tricky. Monosaccharides and disaccharides can both be called simple sugars. So it's very common for people to refer to sucrose, for example, as a simple sugar. So one or two subunits make up a simple sugar. Now, the next category of carbohydrate is the oligosaccharide. That term translates to few sugars. Oligo means few. Now, what do we mean by a few? We actually have a definition for that. Any carbohydrate that has between 2, or I should say 3, because of course 2 would be a disaccharide. Any carbohydrate that has between 3 and 20 sugar units within it is called an oligosaccharide. We don't talk about oligosaccharides very often because they are not typically used by microbial cells or really any cell for the kind of metabolic reactions that we learn about. Oligosaccharides are much more commonly found on the outside surface of cells. Those are the carbohydrates that are attached to the outside of the membrane and help give cells their identity. Finally, we have polysaccharides. That's any carbohydrate that has more than 20 sugar units. A couple of the very common ones are starch and glycogen. Starch is found in plants. Glycogen is found in animals. Both of those big molecules are made up of glucose, just glucose. In fact, cells use starch and glycogen as storage forms of glucose. You store glucose, for example, in your skeletal muscle as glycogen. All kinds of glucose molecules all bound together to make up the glycogen molecule. Plants store glucose as starch. Now, when you hear the term complex carbohydrate, and that's another very common term in everyday language, that's referring to a polysaccharide. So a simple sugar is a monosaccharide or a disaccharide, and a complex carbohydrate is a polysaccharide. Now, two other terms that I'll review really quick from the metabolism lectures. These terms, catabolic and anabolic. You know, when you use the word metabolism in common everyday language, again, with people who might not be students of science, people think this word metabolism is very hard and difficult, and it must mean something sort of secretive and hard to understand. All that word means, all metabolism means, is the sum of all of the chemical reactions that take place in every one of your cells every day to keep you alive. That's your metabolism. All right, so it's nothing special or hard to understand. It's simply a word to describe all of it, all of the reactions that are going on in every cell in your body to keep you alive. And it would also apply to all of the chemical reactions that go on in one little microbial cell every day to keep it alive. Now, the simplest way to talk about metabolism, because again, a whole lot of different reactions. The simplest way to talk about it is to divide metabolism into two groups. One group is catabolic reactions, and one group is anabolic reactions. That's the simplest way we can divide up metabolism. Catabolic metabolism, anabolic metabolism. Catabolic, remember, refers to reactions that break things down. Anabolic metabolism refers to all of the reactions that build things up. Catabolic reactions break things apart. Anabolic reactions build things up. And all together, the catabolic and the anabolic reactions make up your metabolism. So today we're talking about carbohydrate catabolism. We're talking about how cells break down carbohydrate. What are the reactions that microbial cells are using, and your cells, by the way? What are the reactions that they're using to break apart carbohydrate? And remember, in living cells, all living cells, carbohydrate is the molecule that's being used to produce ATP. Let me say that again. In all living cells, carbohydrate is the macromolecule that is used to produce energy. And by energy, I mean ATP. It's really that simple. And it gets even simpler when you know that there's one carbohydrate, and it's a monosaccharide that's at the center of all carbohydrate catabolism, and that's glucose. Every single living cell on this planet uses glucose in some way to make energy. Some of us break it apart through cellular respiration and oxidative phosphorylation. Some of us do that aerobically. Some of us do it anaerobically. Other cells can ferment. Fermentation is always anaerobic. And we're going to be talking about fermentation in more detail on Wednesday. All cells on this planet use glucose in some pathway to build ATP. See, it's not as complicated as it seems. The hard part is learning the pathways. That's the really hard part. So Carrie's got a comment about protein giving cells their identity. So there are proteins that are exposed on the outsides of cells, Carrie. You're absolutely right. Remember, some proteins embed in the outer membrane of the outer monolayer of the membrane. Some proteins actually go through both bilayers. So there are absolutely proteins jutting off the surface of cells. And those proteins have all kinds of jobs. Some of them are transporter molecules to help things come in and out of cells. Some of them are actually enzymes. Some of them are receptors. Proteins play a lot of jobs. There's one kind of protein that we call a glycoprotein that gives cells their identity. So you're right, it is a protein, but it's a protein that has an oligosaccharide attached to it. That's why it's a glycoprotein. That term glyco refers to carbohydrate. And each individual cell type has their own signature oligosaccharide. So when the immune system comes by, as it does, and reads the surface of cells, it's looking for those glycoproteins. And your glycoproteins, your immune system will see and say, oh, these belong here. These belong in this body. But if my glycoproteins were in your body, your immune system would say, uh-oh, these don't belong here, let's destroy them. So you're right, it is a protein, but it's a special kind. It's a glycoprotein. And it's that oligosaccharide molecule that's attached to it that gives it its identity. Good question. Connie's saying that she remembers catabolism, breaking things apart, and cats, the word cats, because cats like to destroy things. Oh, I love that. I love that. I use cats a lot in memory devices. For example, I always remember that the difference between a cation and an anion. Cations, of course, are positively charged, and anions are negatively charged. I remember that I feel positive about cats. So that's my little memory device to associate cations with a positive charge. But I love that one, that cats are destructive. I love that. I'm going to use that. And then Lacey talking about a professor or a teacher she had once that talked about anabolic steroids as a way to remember anabolic metabolism. And that's a great memory device. We all know that an anabolic steroid, remember, these are those very dangerous performance enhancing compounds that sometimes some athletes use in order to build muscle, right? So if you can think of an anabolic steroid with the word anabolic, you'll remember that anabolic catabolism builds things and catabolic metabolism breaks things down. That's good. Positively charged. Oh, Connie, you got me laughing. Yes, cations are positively charged. I can't help it. I can't help it. I love those things. All right, I'm going to jump back in. So one of the things that we're going to be looking at today as we talk about carbohydrate metabolism is the enzymes that are involved in the breakdown of certain carbohydrates, and specifically for us today, starch. Microbial cells, like all cells, contain a whole lot of different enzyme types, including what are called hydrolytic enzymes. Now, if you break down that word hydrolytic, it means water breaking apart. So a hydrolytic enzyme is an enzyme that's capable of breaking something apart in the presence of water. And microbial cells have several different hydrolytic enzymes to help them break down carbohydrate, not that's inside of them, but that's outside of them in their environment. Remember, microbial cells are single-celled organisms. And so they have to get nutrients from the environment. They break down big complex molecules like carbohydrates by secreting enzymes out into the world. Now, there are two categories of hydrolytic enzymes that I want us to be familiar with. One are exoenzymes and one are endoenzymes. So an exoenzyme is an enzyme that gets secreted out into the world, out into the environment in order to digest something, to break something down. Now, what a microbial cell has sometimes as an exoenzyme, excuse me, all I have to do is glare at them, and sometimes that'll stop them from barking. Girls. What microbial cells have is an enzyme called amylase. Now, you and I also have amylase, don't we? We have amylase in our mouths even. And what amylase can break apart is that molecule starch. So remember, starch is a polysaccharide. It's a plant molecule, plant-based, and it's simply built out of lots and lots of glucose. When there's starch in the environment, certain microbial cells, not all of them, but certain microbial cells will secrete amylase into the environment and break that starch down. Then they can simply absorb the glucose. Okay? Now, the other category of hydrolytic enzymes are endoenzymes. That term endo just means within. Exo means outside of, and endo means within. So there are some hydrolytic enzymes that stay within microbial cells, and they're there to digest certain molecules inside of the cell. Amylase is an exoenzyme. So let's talk about this starch hydrolysis test. This is a very commonly used test in microbiology labs, again, because it tells us something about a bacterium that we need to identify. One of the tests that we can run is the starch hydrolysis test to find out whether or not this particular bacterium can secrete amylase into its environment. So what you're looking at up here on the upper left, this is what a starch hydrolysis plate looks like. Very boring, right? Very, very boring. It's just a basic basal medium with this sort of a beigeish color, but it has starch added to it. Now, the other thing you can see is this big bottle of something and this plate right here, which looks very different. To run the starch hydrolysis test, what we do is we streak cells across the surface of this starch plate, and we put them in the incubator and we let them grow as usual. And then when we take them out of the incubator, we really do the work of this test. What we do is we add iodine onto the surface of the plate. We're just going to dribble iodine onto the surface of this plate, and it will be taken into the agar. Remember something about iodine. We talked about this with the gram staining. Iodine is a mordant, and it's really good at penetrating through materials. So it will penetrate through the agar. And what's going to happen is anywhere that there's starch, we're going to see that the starch media turns this black color. Some people see it more as a bluish color. I see it as a black-brown color. Your eyes may be a little different. Anywhere that there's still starch here, we're going to get the black color change. So here's a starch plate before any cells have been put on it. And when you put iodine on it, the whole plate turns black. It's going to bind to the starch, and you're going to get that black color change. Now, take a look at the bottom image. On both of these plates, there's a linear streak of cells. So you don't have to use any kind of fancy streak technique on a starch hydrolysis test. You don't need to do a quadrant streak. You don't need to use spread plating. All you have to do is get some cells onto the plate. And what this person has done is just spread them in a straight line with their inoculating loop. And remember, we're spreading the cells on the plate when it looks like this. And then we're going to incubate those cells and let them grow. So you can see a line of growth over here, and you can see a line of growth over here, right in the middle of the image. Now, look at the difference in the two plates. Obviously, iodine has been added. Everywhere that there is starch, the agar turns black. If there's no starch, the agar will stay clear. And I do mean clear. There's a piece of paper behind this plate on the left, and the paper has writing on it, and you can read the words right through the plate. No black staining has occurred here because there's no starch here. And the reason there's no starch is because the cells that are growing right here in the center have secreted amylase. And the amylase broke down the starch that's right around the cells. What did it break it into? It broke it into glucose, because that's what starch is made out of. So if I were to take a little chunk of the agar off this plate where it's clear, if I were to take a chunk of the agar and analyze it, it would be loaded with glucose. There's starch out here, there's starch out here, but there's no starch around the line of cells because it's been broken down into glucose. Very different over here. There's cells growing here. There's cells growing, but all of the agar has stained black. And that means that these cells have not broken down any of the starch. These cells must not have the ability to secrete amylase. This is a negative test, and this is a positive test. Any questions about that? This is one of the many, many different tests that we can do in the laboratory on a bacterium that we need to identify. Remember, we're usually not completely in the dark. We usually have at least a short list of possible candidates because we know where our unknown samples are coming from. Sometimes they're coming from patients who have a particular illness, and sometimes they're coming from a particular environment. So we know the usual suspects. Now we just have to figure out which one it is. So we pick and choose which tests we're going to run according to what we think might be the identity of this unknown bacterium. And one of the tests that we can use is the starch hydrolysis test, because only certain bacteria can secrete amylase. Okay. The other test we're going to talk about today is this test called the OF test. And this is a very complex test to interpret. Another really big sort of basic question that we can test or ask about a microbial cell is, what is it doing with glucose? How is it using glucose to make ATP? Because all cells use glucose in one way or another to make ATP. So one of the really fundamental tests we can do is this OF test, because it will tell us whether a bacterium is oxidative or fermentative or both. It tests whether a microbe uses oxidative catabolism. And remember, when we talk about oxidative, this has nothing to do with oxygen. The word oxidative comes from redox or oxidation reduction reactions. Cellular respiration is a long series of oxidation reduction reactions. So that term oxidative is referring to cellular respiration. So some microbes will be oxidative. They will use glucose and cellular respiration to make ATP. Other microbes are fermentative. Some microbes are both. And in all cases, glucose is at the center of that. Sometimes microbes don't have direct access to glucose in its pure form. Sometimes they only have another carbohydrate or another simple sugar, a disaccharide, for example. But remember, disaccharides in cells tend to contain glucose. So sometimes, for example, a microbial cell will have to use lactose to get its glucose. Sometimes it'll have to use maltose to get its glucose. But it's going to break that glucose down in some way to make ATP. Now, an important thing to remember about oxidative catabolism is this. It can be either aerobic or anaerobic. Aerobic cellular respiration uses oxygen as the final electron acceptor molecule at the end of the electron transport chain. Anaerobic cellular respiration uses something else. And as we talk about in the lecture about cellular respiration, we're typically talking about nitrate or sulfate, and the other one that's very common is carbonate. Carbonate. So microbes that use anaerobic cellular respiration use either nitrate or sulfate or carbonate at the end of their electron transport chain as their final electron acceptor. I told you lots of language today. Now it is important to remember, and that's why I've got it in yellow highlight here. The OF test only tests for aerobic cellular respiration. Okay? The oxidative part refers to respiration and specifically to aerobic cellular respiration. We always have to remind ourselves that microbes are very diverse. And there are bacteria that can only do cellular respiration, either aerobic or anaerobic. But there are also bacteria that can switch back and forth with fermentation, and there are some microbes that can just ferment. They're very diverse. The microbes that only ferment, the ones that take glucose and ferment it to get their ATP, we refer to those as pure fermenters. But when we talk about the majority of microbes, they're able to switch back and forth between pathways. There's more yellow highlighter on this slide because there are more really important points as we start talking about the OF test. Key point number 1, anaerobic cellular respiration is not the same thing as fermentation. Please put a star next to that in your notes. Anaerobic cellular respiration is not the same thing as fermentation. That anaerobic term means that oxygen is not being used. Some other molecule is being used though. So it's still cellular respiration. Let's review this. When I use the word cellular respiration, I'm talking about a combination of metabolic reactions. Glycolysis, pyruvate processing, the Krebs cycle, and then the electron transport chain. At the bottom of the electron transport chain, there's going to be a final electron acceptor. It's going to accept the electrons that have been set free from glucose, and that part is done. The glucose molecule is broken down all the way to carbon dioxide during that long pathway. Glycolysis, pyruvate processing, the Krebs cycle, and the electron transport chain. That's cellular respiration. The glucose is broken all the way apart to carbon dioxide. All of its electrons get harvested, sent down the electron transport chain so their energy can be used for what comes next. And of course, what comes next is oxidative phosphorylation. Remember, oxidative doesn't mean oxygen. It means redox reactions. So oxidative phosphorylation is a process that builds ATP using redox reactions. All of the energy that came out of the electrons in the electron transport chain, all of that energy is used to build a gradient of hydrogen ions. In oxidative phosphorylation, all of the energy from the electrons that were released from glucose is used to build a proton gradient or a hydrogen ion gradient across the bacterial cell membrane. That gradient, those hydrogen ions, they're then going to flow back into the cell through a molecule called ATP synthase. And as the hydrogen ions come through that molecule, and they flow through the center of the molecule, they're going to cause it to spin. It literally moves, it spins. And that mechanical motion phosphorylates ADP and creates ATP. It's a long, long process to take a glucose molecule and make ATP with it. Respiration is the first part, followed by oxidative phosphorylation. It's really complex, isn't it? It's very, very complex. And the language gets hard. But that's what we're talking about. That's what we're talking about. Some microbial cells do it with oxygen as the final electron acceptor. Some microbial cells use a different molecule. Nitrate, sulfate, and carbonate are the most common that you'll find. Each species uses a different molecule. Those processes, though, are anaerobic. They're still doing cellular respiration. They're still doing oxidative phosphorylation, but they're using something other than oxygen. Fermentation is completely different. Fermentation is completely different. It has nothing to do with cellular respiration and the electron transport chain and all of that. Okay, so it's really important that you keep those reactions separate in your head. Here's the second key point. Remember, we talked about oxygen requirement categories. We talked about how we use thioglycolate medium in order to figure out which category a bacterial cell belongs to. Oxygen requirement categories are only useful to us in terms of describing the relationship that a particular species has with oxygen. Okay? Oxygen requirement categories tell us nothing about whether or not a cell can ferment. Let me say that again, because that's another key point. Oxygen requirement categories, and I've got them listed here, they tell us nothing about whether or not a microbe is capable of fermentation. We have to test for that separately. Now, why is that? Well, third key point. Fermentation doesn't require oxygen, even in aerobic organisms. Now, that seems strange. Aerobic organisms, the organisms that require oxygen to make ATP, they're using that oxygen at the end of their electron transport chain. That's what we mean. When we call them aerobic, we're saying they require oxygen to make ATP in that process called cellular respiration. Fermentation is different. There's no place in fermentation where an oxygen molecule is involved. In respiration, the oxygen molecule sits at the bottom of the electron transport chain and receives all of those electrons. But in fermentation, there's no oxygen involved. So even in you and I, and we are certainly aerobes, you and I are capable of fermentation under certain conditions. We can do lactic acid fermentation. It's an anaerobic process. There's no oxygen involved, even though you and I do require oxygen. Our oxygen requirement category is obligate aerobe. But we're not using that oxygen when we ferment. Okay? So keep the oxygen category and fermentation separate. They're not related to each other. Okay? All right. Let's switch gears now and talk a little bit about this OF medium. This is a basal medium. Remember what we mean by that. It's a chemically defined medium. The list of ingredients are all known. And what can grow in a basal medium? Any kind of microbe that is not particularly fastidious. Remember that word fastidious? Non-fastidious microbes can grow in basal media. So we take this basal medium and we add a lot of carbohydrate to it, and particularly we add glucose. And then we put a small amount of that molecule called peptone in there. Now remember what peptone is. Peptone is partially digested protein. So we put a lot of glucose in, we put a small amount of peptone in. Why do we put the peptone in? Well, remember, there are going to be some microbes that aren't going to be able to grow in either oxidative or fermentative ways in this test. There are going to be some microbes that aren't really going to grow for us in oxidative or fermentative ways. And that's because this test only looks at aerobic oxidative pathways, aerobic cellular respiration. So it's testing two things. Can the organism ferment glucose? Can the organism use glucose for aerobic cellular respiration? There are going to be some microbes that they can't do either one of those. There are going to be some microbes, for example, who can only do anaerobic cellular respiration. If you put that organism into an OF tube, they can't do it. They can't do aerobic cellular respiration. So what's left for them? They can't use that glucose to make energy. They just can't do it. But because of the peptone that's in there, they can at least survive temporarily in that tube. So it allows them to survive the peptone, but they can't do aerobic, oxidative catabolism of glucose. They just can't do it. And listen, there are going to be other organisms that can't grow at all for us, even with the peptone. And that's because it's a basal medium. Remember, every once in a while, we run into fastidious bacteria. They have very special nutritional requirements. And if we can't meet those nutritional requirements, they can't grow. So sometimes bacteria just won't grow because the media doesn't give them all the nutrients they need. I told you it was a complex test. The other thing that's in OF medium that we need to be familiar with is an indicator dye. This one is one we haven't talked about yet. It's called bromthymol blue. Now, bromthymol blue is going to turn yellow in acidic conditions. We've seen that with other indicator dyes. In acidic conditions, it will turn yellow. It'll also turn blue, though, if it's in an alkaline condition. This is going to be helpful to us because if the organism is breaking apart the glucose, we're going to get a yellow color change. If it's metabolizing the glucose through either oxidative or fermentative metabolism, it's going to turn yellow. If it's not able to do that, but it can metabolize the peptone just to stay alive, it's going to turn blue. So we've got multiple colors in this test that we need to be able to interpret. Now, the OF test is oxidative fermentative. That's what the name means. We run this test in pairs. We use 2 media tubes. One of them is going to test for oxidative breakdown of glucose, and one's going to test for fermentative breakdown of glucose. We leave one of the tubes open to the air, and we take the other tube and we put a layer of oil on top of it, mineral oil specifically. Now, why do we do that? Well, that will keep oxygen out of that tube. That's going to be the tube we use to test for fermentation. The open air test is going to test for aerobic cellular respiration. The oil-covered tube is going to test for fermentation. So you always run this test in pairs. On this slide, you're looking at 3 pair of OF tubes. And I think you can just see that there's a layer of mineral oil on top of the media in one of each pair. You just drip that mineral oil into the tube, and because it's less dense, it will sit right on top. It's going to block the media and prevent oxygen in the air from getting in. We want to keep the oxygen out because we want to create an anaerobic environment. We want to create an environment that will push the microbe to ferment, encourage it to ferment, if it's capable of that. Now, this slide shows us how to interpret the test when we take the tubes out of the incubator. Remember, we're going to put these tubes into, we're going to inoculate them, and we're going to put them in the incubator. We're going to let those cells grow for 24 hours because we want them to metabolize. In fact, we're actually going to let them grow longer in some cases. The media starts off green. This is what the media looks like before we inoculate it. It has a green color. We use our inoculating loop and our best aseptic technique. And we inoculate the tube and we incubate the tube and then we read the tube. So let's take a look at this first pair. The first thing you should evaluate is this color change. I see the color yellow. Let's remember what yellow means in this test. Yellow means acidic conditions. So there's an acidic pH in both of these tubes. And when I get that acidic pH, that signals to me that the carbohydrate, the glucose in there, has been broken down. So that's the first thing I do is I interpret the color. I have two acidic tubes, carbohydrate catabolism. Now I'm going to evaluate what each tube means. The tube that's open to the air is testing for aerobic cellular respiration. If the microbe was able to do it, the tube will turn yellow, and it has. So I've got a microbe in here that's doing aerobic cellular respiration. This tube tests for fermentation. If the microbe is fermenting the glucose, the tube is going to turn yellow, and it has. So I have a microbe here that can do both things. It can do oxidative and fermentative catabolism or breakdown of glucose. Now look at the middle one. Positive result right here for aerobic cellular respiration. This microbe performs aerobic cellular respiration of glucose in the open air tube. In the fermentative tube, I have no change. The media stayed green. So this particular microbe can do oxidative metabolism, but not fermentative. When you cover the tube with oil and you block out the air, it cannot grow. It's not growing in here. That's why the media is staying green. And finally, we do get this result sometimes. We take the tubes out of the incubator and both tubes are green. No change at all in color. What's happening? Well, the glucose is not being catabolized in either tube. It's not doing aerobic cellular respiration. It's not doing fermentation. In other words, that microbe can't do either pathway under the conditions that we have provided for it. What does it mean? Well, what does it mean? Well, what else might it be able to do? It can't do aerobic cellular respiration of glucose to make its ATP. It also can't do fermentation of glucose to make its ATP. Can't grow in those tubes. So what's it doing out in the world? How's it surviving? How is it getting its ATP? What else could that microbe be doing? Type it in chat for me. It's not doing aerobic cellular respiration and oxidative phosphorylation. If it was, the tube would have turned yellow. It's not doing fermentation. If it was, the tube would have turned yellow. Both tubes stay green. It's not even growing. It can't even use the peptone. What must it be doing out in the real world? Remember, there are three pathways for the breakdown of glucose, the catabolism of carbohydrate for the production of energy. Just three. It can't do two of them. It must be doing the other one. Connie's got it. Connie's got it. Good job, Connie. Maybe it does anaerobic cellular respiration. So why can't it do it? Why can't it do it in one of those tubes? It can't do it because it doesn't have its final electron acceptor. Remember, the OF test only tests for aerobic cellular respiration, that's the open tube, and fermentation, that's the oil covered tube. It doesn't test for anaerobic cellular respiration. That's probably what that organism needs to do, and it can't do it, so it can't grow. Now, we've got 3 pairs of tubes here. We've got the mix and match of colors. We can interpret those, but there's no blue tubes here. Remember, I said one of the things that happens sometimes is the media will turn blue. Remember what a blue tube means. A blue tube means growth without the catabolism of glucose. If you see blue, it means that the bacterium is growing in there, but it's barely surviving because it cannot break down the glucose under these conditions. If you see a blue tube, you've got an organism that is desperately trying to survive right now. It's using that peptone molecule, that partially digested protein, in order to just survive for a little while longer. It cannot break apart the glucose in the tube using aerobic cellular respiration or fermentation. So it's dying, but it at least can make use of that peptone. So remember, blue is an option. It happens in this test sometimes. So when you interpret the OF test, you have to do 2 things. First, evaluate the color changes, if any of them have occurred. Second, compare the open tube to the oil-covered tube, because the open tube is the tube that's testing for aerobic cellular respiration. The oil-covered tube is testing for fermentation. Start with the color change. If it has turned yellow, it means acidic conditions. That organism is growing in the tube and it is breaking apart the glucose. That's what yellow means. The oil versus the open tube will tell you what pathway it's using. But yellow tells you acid, breakdown of carbohydrate is occurring. If the tube stays green, because this medium starts off green, that means no change is going on, no growth is occurring, no metabolism of glucose is occurring, no metabolism of peptone is occurring, no growth is going on in that tube. And if the tube turns blue, now you've got alkaline conditions. The pH is high. There's growth going on in there, but no metabolism of the carbohydrate, no metabolism of the glucose. That organism is using that little bit of peptone just to survive, just to get by a little bit longer before it dies, essentially. Now, one of the things that I want us to start thinking about when we talk about glucose catabolism, when we talk about oxidative metabolism and fermentative catabolism, is where does the acid come from? These tubes are turning yellow and acid is being produced. Where the heck is this acid coming from? Let's talk about the fermentation tube first. Remember, during fermentation, glucose is going to be converted into pyruvate. That's the process called glycolysis. That's still occurring. But the pyruvate is going to be converted into some combination of products. It's going to be converted into acid or alcohol or gas or some combination. any acid that gets produced in that fermentation tube is going to turn the color to yellow. There are lots of different fermentation pathways. They're not all the same. And there are different acid products that can be made during fermentation. It depends. It depends on the substrate and the organism involved. It depends. Notice I've got a little asterisk here though. In the fermentation test, if the organism ferments, regardless of what fermentation pathway it's using, if it ferments, the tube's going to turn yellow. And that's because of this. In all fermentation, and there are many different pathways of fermentation, at least one acid gets made. And that acid is pyruvate. During fermentation, the first thing that happens is glycolysis. Glucose gets broken down to pyruvate. Another word for pyruvate is pyruvic acid. So all fermentation pathways produce pyruvate, at least. And some of them produce even more acid as a product molecule. So regardless of the type of fermentation going on, if an organism can ferment, the fermentation tube will turn yellow. Okay? There are lots of fermentation pathways. It's amazing how many different fermentation pathways are available in nature. You and I use lactic acid fermentation when our cells ferment. There are bacteria that can do that one too, but there are many others as well. Sometimes the product molecule is an acid. Sometimes it's an alcohol. Sometimes it's a gas. Sometimes it's a combination of those. But there's always pyruvate made, regardless of what the product molecules are. Now let's talk about the oxidative tube, the cellular respiration tube. Remember, it's aerobic. That one also turns yellow when aerobic cellular respiration is going on. So where is that acid coming from? Remember what cellular respiration is. It's a series of metabolic pathways that breaks glucose down. Glycolysis takes glucose to pyruvate. Pyruvate processing takes the pyruvate and converts it into acetyl-CoA and carbon dioxide. The Krebs cycle then takes the acetyl-CoA and converts it to carbon dioxide. By the end, you've got carbon dioxide. The glucose has been broken down into carbon dioxide. In a living cell, carbon dioxide gas will combine with water. And guess what it makes? An acid. It makes something called carbonic acid. That's what's turning the tube yellow. If aerobic cellular respiration is going on, the tube's going to turn yellow because the glucose has been turned into carbonic acid at the end of all those reactions. Three colors: yellow, green, and blue. Yellow means acid. Yellow means growth and breakdown of glucose, metabolism of glucose. Green means no growth, no metabolism of glucose. Blue means growth, but no metabolism of glucose. So how come there's a difference here? How come there are some organisms that can grow, but they can't break the glucose down? Some organisms that'll use the peptone and some that won't. What's the difference there? Well, it's a basal media. It's a basal media. Some bacteria cannot grow in OF because it's a basal media. It just doesn't have the nutrients they need. Some organisms can grow in it, and they can break down that peptone just to keep them alive, essentially, because they can't manage to break down the glucose. Very complex test. Very complex test. So do yourself a favor when you look at a pair of OF tubes. First, evaluate the color. What does the color mean? Then think about open air tube versus oil covered tube. What does that mean? Think about the color first. Okay, and you won't get confused. It's a hard test. It's a tough test to interpret. And it's easy, I know, it's easy to get jumbled up in all the terminology. Interpret it the same way every time, and you'll have a better go of it. All right. Are we ready for an experiment? Are we ready to do some OF tubes and look at the results? Any questions so far? All right. Let's take a look at some OF tubes and let's interpret them. We're going to use 3 bugs today. We're going to use our old friend E. coli because we love E. coli. We're going to use another organism we've seen before, Bacillus subtilis. And we're going to use another organism we've seen before, Pseudomonas aeruginosa. Remember, E. coli is a gram-negative rod. We learned from the oxygen category lab that it's a facultative anaerobe. Remember that? Bacillus subtilis is a gram-positive rod. And Pseudomonas is a gram-negative rod. So we have a little bit of a mix here. Think about E. coli for a minute. E. coli is a facultative anaerobe. Let's see if we can predict what E. coli is going to do in this test. First, let's think about facultative anaerobe. Does a facultative anaerobe require oxygen to make ATP? Yes or no? Type it in chat for me. Does a facultative anaerobe require oxygen to make its ATP? Yes or no? Facultative anaerobe. Remember, most bacterial species fall in that category, including our friend E. coli. Does a facultative anaerobe need oxygen to make its ATP? Good job. Type it right in the chat for me. Lacey's got it. Valerie's got it. Elaine's got it. Connie's got it. Marilyn's got it. No, a facultative anaerobe is very adaptive, remember. They don't require oxygen to make ATP, to break down glucose and make ATP, but they love to use oxygen to do that, right? If there's oxygen around, that's when they grow best. They love to do aerobic cellular respiration. If there's oxygen around, they'll do it. That's when they grow best. But if there's no oxygen around, no problem. They'll just switch to another pathway. They're facultative. They're very adaptive. Remember, they grew best at the top of the all the way through the oxic zone in the thioglycolate tube. They grew really well up there, but they also grew in tendrils through the rest of the tube. Very adaptive organism. So what do you think about this E. coli? What do you think is going to happen in the open air tube? Is it going to turn yellow? Is it going to stay green? Might it turn blue? What do you expect to see in the open air tube? For E. coli, our facultative anaerobe. Should E. coli be able to do aerobic cellular respiration? Because that's what the open air tube is testing for. Yes or no? Type it into chat for me. Is that open air tube going to turn yellow? Green? Is it going to stay green? Remember, evaluate color first. What does yellow mean? Lacey's got it. Lacey, say that proudly. Say it proudly. You don't need that question mark. Yes, E. coli is going to turn that open air tube yellow because yellow color tells us acidic conditions, growth, and aerobic cellular respiration is going on. E. coli loves to do aerobic cellular respiration because it's facultative. What about the oil-covered tube? What about the oil-covered tube? Hard question here. Hard question. Can a facultative anaerobe ferment? Yes, no, or I don't know. Can a facultative anaerobe do fermentation? Yes, no, or I don't know. Hard question. Lacey, I love that. The Swiss Army knife of bacteria. All right, here's why that's a very hard question. I don't know. I don't know. I don't know. Remember, oxygen category tells us nothing about whether or not an organism can ferment. Write that down in your notes and put a star next to it. Oxygen category tells us nothing about whether or not an organism can ferment. Oxygen category only tells us about their relationship with oxygen and cellular respiration. We have to test fermentation. We have to think of fermentation very separate from that. Remember, the role of oxygen in metabolism is to sit at the bottom of the electron transport chain. So oxygen category, we're only thinking about respiration. We can't use their oxygen category to guess or predict what's going to happen in fermentation. That's why we have to do this test. So we don't know. We can't predict right now whether or not E. coli is going to ferment. But since we know, as Lacey said, that they are the Swiss Army knife of bacteria, it's a good bet that they can ferment, right? It's a good bet. They're facultative. They're very adaptive. So I would put my money on yes, they can ferment. But you can't tell that just by their oxygen category. All right. We're going to put those 3 microbes into 2 kinds of media. We're going to run that starch agar test. Remember the starch agar? I can't say that today. The plate is just a plain old nutrient media that has a lot of starch in it. We're going to put a simple streak down the middle of the plate or a zigzag streak. It doesn't have to be fancy. We're going to incubate it for 24 hours. We're going to put some iodine on top, and we're going to look for those clear zones. We're also going to inoculate a pair of OF media tubes. One tube we're going to inoculate and leave open to the air in the incubator. One tube we're going to inoculate, and then we're going to cover it with mineral oil and incubate it. We're going to take the tubes out after 24 hours and look for those color changes. Before we look at our results, I want you to take a look at this recipe. This is a recipe for OF medium. Just like all culture media, you can buy OF as a powder and mix it up yourself in the lab, or you can buy it pre-made. If you buy it as a powder, you can read the recipe on the side of the bottle, and here it is. It's a pretty short recipe, isn't it? Peptone, remember there's a little bit of peptone in there. Glucose, of course, that's our carbohydrate. It has a little bit of salt in it. Here's our bromethymol blue, our indicator dye. It also has some other, what am I trying to say? Other minerals in it, other nutrients in it. It has potassium and it has phosphate in it. Very simple basal media. Now, take a look. Are there any final electron acceptor molecules in here that an organism could use to do anaerobic cellular respiration? Do you see any nitrate? Do you see any sulfate? Do you see any carbonate? No, there's nothing like that in here. This media will only support aerobic cellular respiration. Hey, how does the oxygen get in there? Because I don't see oxygen in the list either. Where's the oxygen come from in OF media? Where does the oxygen come from? I didn't see oxygen in the list either. How come organisms can do aerobic cellular respiration in that tube? I don't see any oxygen in that recipe. Where's the oxygen coming from? What do you think? Look at that recipe. I don't see nitrate or sulfate or carbonate, and those are the three most common final electron acceptor molecules for anaerobic cellular respiration. But I also don't see any oxygen in here. And we know that this tests for aerobic cellular respiration. So where's the oxygen come from? You've got it. Elaine's got it. Connie's got it. Valerie's got it. Comes in from the air. Like Elaine has said here, we didn't do anything to stop oxygen from dissolving into that media, did we? We didn't do anything to stop that. If we had put some agar in there, like thioglycolate medium, if we had put some agar in there, we would have prevented oxygen from dissolving into that medium. But in an open air liquid broth, oxygen comes from the air. It dissolves right into the liquid. So that's how it tests for aerobic cellular respiration. It's open to the air. The air provides the oxygen. Good job. All right, let's take a look at our results. These are our starch hydrolysis plates. Here's the E. coli plate after the iodine was added. You can't see the streak, but trust me, there's a streak here. The plate is angled badly for the photograph, but there is a streak of cells growing here. Black, black, black plate. Here's the Pseudomonas plate. You can see the streak of cells much better on this one. And the Bacillus subtilis plate, we streaked the cells in a zigzag on this one. It's a little harder to grow Bacillus. The name subtilis tells you that it's a subtle organism. It's a little bit harder to grow it. So we just streaked more cells onto this plate. We added iodine to all 3 plates. What do you think? Type into chat for me. Which of these organisms secretes the amylase enzyme? Which one of these organisms, if any, secretes the amylase enzyme? What do you think? We don't have the plates held up to a piece of paper, but I still think you can interpret them. Good job. The bacillus. There's a clear zone around the bacillus streak, right? There's a clear zone. Those cells have secreted amylase, that exoenzyme, and the starch in the media has been broken down by the amylase. So if I were to scoop a little of the agar out in the clear areas, it would be loaded with glucose. The rest of the plate still has starch in it, which is why it's black. The amylase can only travel so far when it gets secreted, but it breaks the starch down and it makes glucose available, and those cells will absorb that glucose. So Bacillus subtilis can secrete amylase. It's one of its traits. E. coli doesn't do that, and neither does Pseudomonas. Neither one of those organisms is able to secrete amylase. They just can't. They don't have it. But Bacillus can. All right, let's look at our OF tubes. These 2 tubes are for E. coli. I think you can just see I've got the names down in white. These 2 tubes are Pseudomonas, and these 2 are Bacillus subtilis. And I put the reference tubes down here that we looked at earlier. Evaluate color first. Then compare the open air tube to the oil covered tube to figure out your pathways. Let's do E. coli. How would you interpret the E. coli tubes? What can E. coli do or not do? Can E. coli use oxidative catabolism of glucose to make ATP? Right, type it right into chat, yes or no. Can E. coli use oxidative catabolism of glucose to make ATP? Yes, 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 yes. How do I know? The yellow media tells me an acid is in that tube. The yellow media says acid pH. That's a positive test result. Open air means oxidative or cellular respiration is occurring. The acid comes from the carbon dioxide, carbonic acid. Good. What about the oil covered tube? Can E. coli use fermentative catabolism of glucose? Yes or no? Can E. coli use fermentative catabolism of glucose? In other words, can E. coli ferment glucose? Yes, yes, because the other tube is yellow too. Remember, if fermentation occurs in the oil-covered tube, the tube's going to turn yellow, regardless of what the end products are. Regardless of what the end products are. The end products could be more acid, alcohol, or gas. We're not evaluating the end products. We're just evaluating whether or not it can do it. If it's yellow, it can. E. coli is the Swiss Army knife of microbes. It can use both kinds. It's very adaptive. Let's go over to the bacillus tubes. Can Bacillus subtilis use oxidative catabolism of glucose? Yes or no? Can Bacillus subtilis use oxidative catabolism of glucose? Yes or no? We do have an interesting situation here, don't we? Some green, some blue. What does that mean? No, no, it can't. Evaluate the color first. A positive test would be a yellow tube. This tube is definitely not yellow. It's some green, some blue. Is there any Bacillus subtilis growing in that open air tube? Yes or no? Is there any Bacillus subtilis growing in that open air tube? Yes, Valerie, you've got it. Yeah, there's some E. coli growing in there. How do I know? Because at least part of that tube turned blue. It's not growing well, is it? It's not very happy. It's having trouble making ATP. It's not growing well. It's not going to survive long. But it can use alternate metabolic pathways, at least temporarily, to survive by using that peptone. And we don't talk about these alternative pathways that involve protein because that is not primary metabolism. All cells use carbohydrate to make ATP as their primary metabolism. But it is possible to survive at least a little while off of protein. The blue bit, I like that, Connie. The blue bit, that's right. That's where it's growing and breaking down the peptone. Does anybody have any sense of why the whole tube isn't blue? We said blue means alkaline pH. And the alkaline pH comes from the breakdown of peptone. Why isn't the whole tube blue? That's a really tricky question. It's more about chemistry than microbiology. Any idea? Think about that one for a minute while we look at the other tubes. Here are our Pseudomonas tubes. Oh, wait, before I do Pseudomonas, silly me. Look at the oil-covered tube for Bacillus subtilis. Is Bacillus subtilis fermenting the glucose in this tube? Is the Bacillus subtilis fermenting glucose in that oil-covered tube? Yes or no? Type that into chat. The oil-covered tube is green. What does green mean? No change, right? No change. No growth. There's no growth of cells in there because the media hasn't changed. No pH change at all. No, it can't ferment the glucose. Okay? Can't do it. All right, now take a look at the Pseudomonas. We'll start with the fermentation tube. Can Pseudomonas ferment glucose? Compare it to the Bacillus. Those look very similar, don't they? Pseudomonas is not fermenting that glucose. If Pseudomonas was fermenting that glucose, the pH would have changed. It would have become acidic and the tube would have turned yellow. I don't see that. I see green. No fermentation. Look at the open air tube though. Another weird tube. 2 colors. In the open air tube, we've got yellow at the top, green at the bottom. It's very similar to this one with blue at the top, green at the bottom. What the heck is going on here? I see yellow, so I know that there's acid in here because only an acid pH can turn this tube yellow, but it's not yellow all the way down. What the heck? That's a positive test result. There's yellow. That's a positive test result, but it doesn't look like what we've seen so far. The whole tube isn't yellow. There's only yellow up at the top. It's very strange, isn't it? This is a little more about chemistry than it is about microbiology, because this is about how many cells are in that tube. In the Pseudomonas tubes and those Bacillus tubes, there aren't very many cells in there yet. Both of those organisms tend to grow slowly in OF media. Remember, Bacillus subtilis is kind of hard to grow anyway, hence its name. But Pseudomonas will grow really well for you on some kinds of culture media, but in OF, it grows very slowly. So there aren't that many cells in there. That's all. They're metabolizing the glucose through oxidative catabolism, but they're doing it slowly in terms of building up the acid. There's only a few cells, so there's only a little bit of acid so far. So only a portion of the tube has turned yellow. Same thing is going on in that blue Bacillus tube. There's only a few cells, so there's only a little bit of alkaline product being made so far. If you put those tubes back in the incubator for another day, maybe 2 more days, the whole tube will change. It's all about cell number. So we got slow growing cells in here. Slow growing cells in here. In fact, take a look at this slide. We've got 1, 2, 3 pair of OF tubes. Notice that all 3 of the fermentation tubes are green. There's nothing going on in there. But look at the open air tubes. Little bit of yellow, little bit more yellow, all the way yellow. These have Pseudomonas in them. This one incubated for 24 hours. This one incubated for 48 hours. This one incubated for 5 days. It took 5 days for that tube to turn yellow all the way. It's only because Pseudomonas doesn't grow well in OF. It'll grow, but it doesn't grow quickly. So you just have to know that about that organism. And if you see that it's only partially yellow, you can say to yourself, oh, it needs to go back in the incubator. But I already know it's positive because it turned yellow, and it's only going to turn yellow if an acid is being made. And in an open air tube, that acid comes from cellular respiration. First, evaluate the color change. You've seen this slide before. Yellow means acidic, growth is occurring, catabolism of carbohydrate. Blue means alkaline, growth is occurring, but there's no metabolism of carbohydrate. That one is metabolizing peptone. Green means no growth, no change in pH, no growth, no metabolism going on. That's what you look at first. Then compare the open air versus the oil covered. That's going to tell you the pathway. If it's yellow in the open air tube, it's catabolism of glucose by oxidative pathways, cellular respiration, aerobic cellular respiration. If the oil covered tube is yellow, that's catabolism of carbohydrate, catabolism of glucose through fermentation. Do the color, then do open versus oil covered. Here are results again. Positive, positive, positive. Carbohydrate catabolism is going on. This one is aerobic cellular respiration. This one is fermentation. This one is aerobic cellular respiration. So when you go back through your notes for this, especially when you're getting ready to do the lab homework questions later this week, you want to be able to answer these questions. Of the 3 organisms that we looked at today, which ones are secreting amylase? We talked about that. Which ones of those 3 can aerobically oxidize glucose, in other words, using aerobic cellular respiration? Which ones can ferment glucose? Which ones can grow but are unable to break down glucose in this OF medium? These are our plates for the starch hydrolysis test. These are our tubes for the OF test. You've got all of this in here for yourself when the time comes later in the week to do your lab homework. Now, you can't answer the fermentation lab questions because the fermentation lab is on Wednesday. The OF test only tells us if an organism is capable of fermentation. But I told you there are many different pathways of fermentation. And it turns out that different organisms can ferment different carbohydrates. They're all going to be simple sugars for the most part, but different bacteria can ferment different carbohydrates. Some of them can ferment lactose. Some of them can ferment sucrose. Remember coliforms. Some of them can ferment mannitol. Remember Staph aureus. There are lots of different fermentation pathways. We can't talk about them all, but on Wednesday, we'll look at a test that helps us see which types of fermentation are going on in terms of which sugars bacteria can ferment. And that's coming up on Wednesday. All right, guys, don't forget to get your lecture work done and your lecture quizzes. I will see you on Wednesday morning. Try to stay dry today. Good job, good work today. I know it's hard material. And we'll see you next time.